For our second lecture, I'd like to introduce you guys to chemistry. Now, I understand that many of you guys have taken chemistry. Hopefully, you've already taken chemistry here at the university. But if not, I want to emphasize to you that the title of this course is Molecules, Cells, and Genes. We're going to start at the molecular level, individual molecules, and it's chemistry that governs this interaction. I'm going to expect you to be able to describe electronegativity, because electronegativity is going to determine bond types and how those bonding types in larger molecules influence the interactions of those molecules with the water around them. And by the way, if you didn't know this, you're a big bag of water. So we care very much as living things about how molecules interact with those waters. We're also going to connect this to acidic and basic properties. And although I'm not going to be going through them in lecture, your textbook does a fantastic job of connecting acidic and basic compounds with the process of buffering, which is absolutely essential for living things. So what I'd like you to do is pause the video here, log into Socrative if you haven't already done so. This continues on from our previous lecture. And start by reading through this experiment and identifying these components. Again, you are not graded on accuracy. I actually don't care if you get these right, but I'm going to be asking you questions like this on the exam, so you probably care whether or not you get them right. And if you can't do this question in under a minute, all 4 of these, then you're not going to be at the rate, the speed that you need to be at for our lecture exam. So put your notes away and try doing it out of your own noggin. Pause the video and restart it when you're done. All right, so here's a real experiment published in 2019 looking at the influence of gum disease on Alzheimer's. Our independent variable is going to be something that is changed by the researchers. In this case, it's whether or not the bacteria were introduced, since those bacteria are the root cause of gum disease. The dependent variable is what these researchers measured, which is the time to complete the maze, as a measurement, a proxy for this brain disease. The variable that researchers are controlling for with their control group is damage to the gums, meaning the amount of damage that's being caused. That's why even though the control group doesn't get bacteria introduced, right? That's our independent variable, whether or not the bacteria were introduced. By the way, a variable has to have at least 2 sides of the coin. So if you say bacteria is a variable, that's a noun. But that variable that we're going to be controlling for is by making sure that damage to the gums is caused in both groups. Now it becomes a control because it's kept the same. My last question to you is, can a hypothesis ever be proven true? And hopefully you told me that the answer is no, that it can only be supported or unsupported because it's impossible to account for every single variable. Now, our goal today is to really dive into the process of chemistry and connect that to the process of biology, the idea that living things need chemistry. And over the course of the semester, we're going to look not only at chemistry, but as we get into cells and metabolism, we're going to look at the laws of physics as well, because those also govern how our cells as living organisms survive. So when we talk about chemical bonds, we're talking about attractions between atoms. These are typically due to the sharing of electrons. When we share them, they're called covalent bonds. And I'm going to show you that there's an important distinction here. You'll never write the word covalent alone. You will only identify bonds as polar covalent or non-polar covalent, because these are very important. Ionic bonds do also occur, and these ionic bonds are the result of donated electrons. You should be able to see and identify those because these result in ions, which are very important in the kinds of electrolyte solutions and things that are in the body. But these are going to be something that we deal with less because biological molecules are the result of large carbon-containing molecules. That's next lecture. We're also going to look at the influence of these polar covalent bonds on the formation of hydrogen bonds. And I do want to remind you that these bonds, all these different chemical bonds together, is what creates larger things we know as molecules. Hence, this is molecules, cells, and genes. So if a covalent bond is the sharing of electrons, in this case particularly valence electrons between two atoms, that sharing results in a very strong, stable type of interaction. When the sharing is unequal, this is what's known as a polar covalent bond. The cause behind that unequal sharing is when one particular atom in that interaction has a greater affinity for the electrons. This is known as electronegativity. So a more electronegative atom wants electrons more, and a less electronegative atom wants electrons less. So if you have an unequal sharing, an unequal electronegativity in this sharing interaction, then you're going to have electrons spending more time around one of the atoms in that interaction. This is going to result in a partial negative charge. That partial is very, very important. And that partial is going to be something that's written using this little delta symbol here. So for example, oxygen is highly electronegative, and it results in a partial negative charge because electrons will spend more time. Hydrogen is partially positive, and so we write that partial positive here. Now, atoms that are going to be highly electronegative, at least those that are inside of your body, are primarily oxygen and nitrogen. Their electronegativity is on, O and N. Atoms that are weakly electronegative include carbon and hydrogen. And by the way, we consider these two to be equally weakly electronegative. So a carbon to hydrogen bond, even though there's two different atoms on either side, and even though they're not exactly equal, in terms of a biological importance, they're equal enough that we consider these to be nonpolar. Hydrogen bonds are the interactions that form when a partial positive and a partial negative come near each other. That partial positive is almost always going to be a hydrogen. That's why this is called a hydrogen bond. So here's an example of a highly electronegative nitrogen, the partial negative that forms, and a nearby water, which includes a partially positive hydrogen. You'll note that the hydrogen bond is indicated by a dashed line. Now, covalent bonds, which you've drawn in chemistry, are solid lines. And that's because hydrogen bonds, at their core essence, they're very weak and transient. They're not going to be something that stays. This is like a magnet. You can put them together, pull them apart, put them together, pull them apart. Covalent bonds are very strong and stable. And so even though when you put many hydrogen bonds together, they form incredible power, the truth is that an individual hydrogen bond in and of itself is actually very weak. And so it's important to note that covalent bonds are strong and hydrogen bonds are weak. As a reminder, ionic bonds are important because when electrons are donated away, not shared, but donated, they result in full negative or full positive charges. Remember that when we talked about polar covalent bonds, we were doing partial charges. Here we're talking about fully charged atoms because we have an entire electron that's been donated, so you get an entire negative charge. These negatively charged ions are called anions. Positively charged, which are the one that donated the electron away, is going to be a cation. So go ahead and do these next 4 questions on Socrative as an opportunity to test yourself on your knowledge of chemical bonds, polar and nonpolar covalent bonds. So start by telling me which of these are nonpolar covalent bonds. Pause the video here and then do the next 3 questions as well. All right, hopefully you went ahead and did these and you found that they were okay. When we're doing nonpolar covalent bonds, it's going to be very easy. If the 2 atoms on either side of the bond have the same electronegativity, it's automatically nonpolar. A carbon on either side, definitely nonpolar. Carbon and hydrogen are both weakly electronegative, So they're also going to be nonpolar. A polar covalent bond is a sharing of electrons that is unequal on either side, and it results in partial positive and partial negative charges. So our highly electronegative atoms are going to be oxygen and nitrogen. Our weakly electronegative atoms are carbon and hydrogen. So here, a carbon, which is weakly electronegative, and an oxygen, which is highly, that's definitely a polar bond. Here's nitrogen, which is highly electronegative, and hydrogen, which is low. So D and E are both going to be polar covalent bonds. A common misconception is that an oxygen to oxygen bond is going to be a polar covalent bond because you're thinking about electronegativity, and that's great. But even though both oxygens are highly electronegative, because they have the same attraction for electrons, they're ultimately going to be sharing the electrons equally. And so the result of this bond is actually a non-polar covalent bond. Ionically bonded molecules are also sometimes called salts, which is an important component when you think about electrolyte solutions. You're thinking about salt compounds. And a positively charged ion is known as a cation. All right, guys, take a break, sit down, go get a drink, and then come back in my next lecture, and we're going to talk about water.